activation and function of B and T cells. I'm going to talk about primary T cell response as a prototype response. And just for recall purposes, so antigen has been recognized. It has been presented by the antigen presenting cells. And then in context of MSC class 1 or 2, and then T or B cells will come and lock onto antigen presenting cells. And then immune response takes place. We call it effector response. One of such responses is there will be activation of CD4 T cells. So we are going to spend a lot of time on activation of CD4 T cell today. We'll talk about paired interaction between an antigen presenting cells and T cells a little bit in detail. And intracellular events of T cell activation. So remember I said signal transduction. So that is something that's going to be difficult, and I'll go through it, and it's not that easy for you to learn in one lecture. It will need effort on your part, my part, but again, as I said earlier, I will not hold you responsible to know that. But remember that 60 to 80% of the drugs that are coming up are going to target intracellular pathways, to drugs for asthma, drug for AIDS, so on and so forth. So keep that in mind. It doesn't relieve you from the obligation. So it doesn't just takes the stress out for you to prepare for the exam one. And then again, signal transduction in T cell. So we have been talking about primary T cell response, and just to give you a recap on that, and I'm sure you are with me on that, that once an antigen or immunogen comes in the body, so body has, the first step is recognition of antigen. The body wants to find out, do we need to respond or not, right? And then antigen processing. So get used to these terms. And we talked about the antigen presenting cells. I said dendritic cells are the professional cells. We have macrophages. We had thymic epithelial cells, and we had B cells. And in first or second lecture, I did mention about the pattern recognition receptors. These are those receptors. They are there on our cells, and they have an ability to pick up and identify any microbe, may it be a bacteria, virus, or any other microbe, or any other thing that wants to get into the system. So this is a little bit non-specific. And if you remember, I said that they operate through a receptor called Tcell receptor. So these are Tcell receptor. There are quite a few drugs that will target Tcell receptors, especially when we talk of lipopolysaccharide. I'm going to talk about TLR4. So these are some things that you may want to revisit once you go. I also use term naive cells, mature cells, activated cells. So when I use this term, basically what I'm trying to say is that cell is in a different mode of activation, right? So cell was sitting over there in active form, and all of a sudden it becomes active. It's ready to go. So it has to perform a function. And another thing, another word I would use in terms of non-active and active will be immature or mature. So remember, as a rule, our body has tons of these cells, and they are not always in activation mode. They are not ready to go and discharge their ammunition and discharge a lethal hit. They have to go through a process. So there's a due process that is involved, and one such is a pattern recognition receptor. And again, one of you was just asking me why there are so many figures. There was a time we need to make our own figures. So good thing is the new books have their own figures. You just have to pay attention, because for those of us who are visual learners, it will be really good, beneficial that we know what's happening here. Now, let's take an example for gram-negative bacteria, for example, right? So there's a bacteria, it wants to enter our tissue. May it be skin, may it be mucous membrane, you name the tissue. So bacteria gets into the tissue, and then there are cells in the tissue like antigen-presenting cells. One of those cells happen to be immature dendritic cell. And there's a good reason for these cells to be immature, because they aren't mature enough that they have seen that antigen before. So they need to go through a process. That's why it takes time for immune response to set in. Now, they have those receptors. One of the receptors is, I just said, pattern recognition receptors, PAMPs. And in especially dendritic cells, they are the toll-like receptors. And if you remember, I said 1, 2, 3, 4. There was one beautiful picture that we talked about innate immunity. And these are the first receptors that some of the antigens expressed on, say, gram-negative bacteria will come in contact with. So these toll receptors are there like an antennae, and they will come and they will get this signal, and they will attach to a molecule. Let's say it's a lipopolysaccharide in this case. And then also remember that there are some other helping molecules. They are there to make sure that that particular bacteria, you must have heard of terms opsonization of bacteria. Sometimes a sugar coat or a complement attaches. So if you have more than one mechanism of phagocytosis of bacteria, that will enhance phagocytosis of bacteria. So that will increase phagocytosis of bacteria, because the idea is, as I said earlier, your immune system is good enough to fight off any challenge. We don't need any medicine from outside. It's good enough. We are just learning how would an immune system tackle with a bacteria, such a nasty bacteria as gram-negative bacteria, right? And we have phagocytic receptors as well. There are some other receptors as well. There are FC receptors. And all these receptors are expressed on top of those immature cells. They attach with this bacteria. And then, if you remember the other day, I said that once the bacteria is phagocytosed in, and then there's a breakdown of that bacteria, the idea is that antigen has to be processed. Because remember, MSC class 1 or 2 can take a small peptide. So there's a small peptide, 8 to 9, 11 to 17. We talked about class 1 and class 2. So there's a breakdown that is happening in this cell, as you can see, antigen processing. And in the process, the cell is presenting those molecules on the cell membrane on exterior. So there we term we use induction. Like in this case, you didn't see MSC class 2 because there wasn't any need for this cell to express MSC class 2 because there was no antigen attached. So once the signal goes into the cell that I have just phagocytosed a bacteria, and then again that processing takes place, it is a exogenous antigen, and MSC class 2 will be expressed, right, as compared to endogenous when we have MSC class 1. And we have co-stimulatory molecules as well. So this process will activate a dendritic cell, and dendritic cell will do an immune response. Now remember, immune response could be in many different forms. One of the immune response will be expression of these receptor molecules on the exterior of the cell, right? One of the immune response will be to call in T-cell for help. One of the immune response will be to secrete some chemokines so that they become visible, they become noticeable, and they can go where they need to go, right? And one of such responses by this whole process of antigen presentation is they start secreting growth factors. So they are growth factors. For example, if I was to tell you that the message goes from antigen presenting cell to a T cell, and messages we need to expand. So there is a proliferation. Those of you who have had experience in working in lab and hospital, and if they fear that you have infection, inflammation, so they will go for the blood test. There are many things they will do. One of the things that they would do in the blood test will be, they will look at your profile of blood cells. Okay. Now, if they find out there are excessive neutrophils, so this means there's a good reason for body to generate neutrophils because there is an infection going on. If they find out that there are more lymphocytes, like lymphocytosis, this means there is a viral infection going on because body is fighting against that. Right? So there is a message will say, expand a particular cell population. And of course, we need growth factors for that. One of the growth factors in this case is IL-12. And these are called interleukins. They are called cytokines. There are many names for them, but they are very important. For example, one of the cytokines is gamma interferon, IL-1, IL-2. As we've been discovering them, we gave them a name. IL-1, IL-2, IL-3, IL-4, right? So it's like Illinois Route 2. Illinois, I actually was going to Wisconsin, I think it was Wisconsin, and I said, huge IL-2. I said, oh my goodness, what is that? It's not interleukin, so it was route IL-2. So maybe Illinois people can learn it better, I believe, because most of the routes are going to be labeled. But IL-12 is a growth factor. Right? So this is a growth factor. Now, this next step you need for the whole process is you want this cell to migrate because it has just picked up an antigen. Because remember, tissues are not the places where we have the factory of production of T cell. The factory and synthesis of T cells is taking place in thymus is in the beginning, right? In the intrauterine life, or maybe the early part of the year. Bone marrow is where progenitor cells are made. Basic concepts. Lymph node. Lymph node. I mean, you can see. Answer is already. It is a lymph node. It says over here, lymph node. Okay. So this cell has to migrate. And if you remember the anatomy of a lymph node, it says lymph node over here. And you will see that happening many times. I'm going to even say, okay, I'll give you 2 credit if you answer me this question, and the answer will be there up front. You just have to pay attention. So this cell carrying an antigen moves to lymph node, and then there it becomes mature. You see now it becomes mature, and then it presents that antigen, which has already been broken up, to a CD4 T cell. Why should it present to CD4 T cell and not CD8 T cell? Why would this dendritic cell present an antigen to CD4 cell and not CD8 cell? Because it just expressed class 2. It is presenting an antigen in context with class 2. So class 2 antigen can only be seen by CD4. If it had made that choice, and I just told you because this is an exogenous antigen, bacteria, it has to be endogenous antigen for the class 1. These are the questions I'm going to ask you, as simple as that. So you can see the whole issue, the whole sequence is there's an immature cell, interacts with the bacterium in the tissue, it processes bacterial protein, there's an upregulation of MSC class 2 and co-stimulatory molecules, and then it comes and sits in T cell area. and that's where T cell proliferation takes place. Remember, I said if there is an injury over here and you have lymphadenopathy, you have swollen lymph. The reason is because this is expanding. Okay? Now, also remember, it may look simple to you, but in immunological terms, every movement that takes place, every processing in this processing plant goes in a very regulated way. So there is a receptor that will be expressed on this dendritic cell, like chemokine receptor, to make sure that this cell will bring it over here and present in the lymph node. So there is very, very precise movement of these cells. So they don't get lost. They are really being signaled and sequenced towards a particular path that will mount a good immune response. Okay. We just talked about a antigen presenting cell talking to a CD4 T cell, right? It may sound familiar to you because I've discussed that earlier as well. So a peptide is broken, that's what you saw. It's put on the top of MSC class molecule, and then it's presented to a T cell, and we have discussed that. And then again, same story, MSC class 2 and CD4 T cell. And also remember, one of the co-stimulatory molecules that was expressed in the last slide, does anybody remember what was the name of that co-stimulatory molecule which was expressed on dendritic cell? Sorry, louder. Co-stimulatory molecule that was expressed on dendritic cell when it also upregulated the expression from MSC class 2, B7. If you notice, it said B7. So B7 is a very important co-stimulatory molecule. And then again, adhesion molecules to make sure that the cells move in the direction they need to move. Okay? So that we already had this discussion. So that will be a little bit a recap of what I already told you. A antigen presenting cell now is talking to a T cell, because this basically will set up activation of T cell. And remember, if I was to come up with a drug or a medicine where I need to stimulate activation of T cell, I want to know what is this talk going on. Because if I can figure it out, what are the stimulants, what are the inhibitors, how this conversation between two cells is taking place, because that will set up the tone for the immune response. Okay? If, for example, if you have a viral infection and there is no lymphocytosis, there are no T cells, you will have a problem. And that may happen when you have recurrent viral infection and you may have some other immunodeficiencies. So let's take it one by one. There is a MSC class 2, a peptide sits with that, and this will snugly fit with TCR. We already discussed that. TCR has an alpha-beta chain. And then there is a CD4 co-receptor, and there is a place on the side of MSC class 2 receptor where this will dock in. If you go back and look at the previous slides, you will see that. So that's the first step where a paired interaction is taking place. Okay? Now, remember I said the response in terms of immune response could be, the message could be, signal could be, let's make more T cell. Let's start synthesis of cytokines. Let's make enhanced expression of those receptors. So these are all effector or immune responses that we normally see. Okay? One such response I just told you, expression of B7. Remember that? I said what was the co-stimulatory molecule expressed when a dendritic antigen picked up B7. So this B7, this is a co-stimulatory molecule. This is going to enhance the immune response. When it gets expressed, it docks onto a corresponding ligand, which is CD28. So when they lock into each other, so there will be enhancement of signal. And remember, for example, you are hypersensitivity, you are allergic, so probably this enhancement is way too much for you and everything is out of control. So you want to regulate it. And remember, the negative signal for that is you have a molecule which is called CTLA4. It says in hatched because it only gets expressed when the T cell gets activated. There's no need of controlling or inhibiting your speed of T cell activation if there is no activation. So activation has to go up and you have to apply the brake. So these are the brakes. That's why we call negative co-stimulator. Okay? Then CD40 talks to CD40 ligand. Does anybody remember what was the role of CD40 when we were talking about CD40 on T cell? Okay. Remember when I said CD40, I said normally there's a message which would produce antibodies which are either M or D. And now you want to do class switching. You want your body to produce other antibodies like G or E or A and so class switching. So this was an important molecule. One of the functions of CD40 was class switching. The other function of CD40, there are many functions, could be more than one function, is that it produces cytokines. And there's a full lecture on cytokines. But these are important because we give cytokines intravenously. We give you interferon. We give you tons of other cytokines to help the system. Then we have adhesion pairs. They are like cell adhesion molecules. If you remember, very first lecture in physiology, we talk of CAMs, cell adhesion molecules. Cell will go and adhere. So if you want this cell to perform the function, then you need the cell to adhere. Right? So you need expression of these ICAMs and then corresponding, because the idea is if an antigen presenting cell is giving a signal to a T cell and it says, because in order for signaling to take place, firstly they have to really glue together. They have to stay till the signaling transmission takes place. So they have to adhere to each other, but it's not a permanent adhesion. It's more of a, remember we talked about electrostatic adhesion, van der Waals forces, and many other forces which are non-covalent. So there's no binding on that. So it can be attach, detach, that kind of a mechanism which is there. Okay, so what happens is that this immature antigen presenting cell, when it picked up the bacteria from the tissue and it reached the lymph node, it became now mature. So there was a naive T cell sitting over there which had never seen this antigen before. So this naive T cell all of a sudden becomes activated and it started showing up, expressing all those molecules which were not there before. Does that make sense? So we will call that activation of T cell, right? And remember this, and actually this is a physical thing. So these cells, antigen presenting cells and the T cells, when they come together and you want to see them under microscope, and you actually see the ligands as well if you want to go for electron microscope. But of course, they come and they dock in. So you can see if they jut out these, stick out these receptors, like if it's a physical thing, so you will see there are some intervening gaps over here, right? So this junction, like if you remember neuromuscular junction and plate, there's usually a gap for you to put the drugs. Remember the drugs go acetylcholine and so on and so forth. So this gap which is there, a junction we call synapse. S-Y-N-A-P-S-E, synapse. So that's the immunological term. I'm going to talk about that. So once all these things are docked together, then the process begins, which is called intracellular activation of T cell. Okay? And that's what I said initially, that those of you who probably are interested in research, and again, this is also my field as well, because those of you will come and do research with me. I look for all these receptors, I stain them with the antibody, I look for T cells, we separate them together, we bring them together, and we look at all those intracellular pathways, and I'm going to talk about all the players. And then again, those of you who do intend to go to R&D, immunology, research, and so on and so forth, so that's going to be their bread and butter. Okay. Now, this is called intracellular events. The other term is signal transduction. So, in the previous slide, that was the initial signal. So that was the first signal. TCR binds to MSC. Second signal was co-stimulatory molecule. Now they cause something on the membrane. Something happens on the membrane, and the signal comes and goes into now inside of the membrane. We call that outside-in signaling. Right? If you remember PI kinase, and if you remember calmodulin, all these kind of in endocrinology. So these are different molecules and the drugs that are going to go. And phosphodiesterol, these kind of things are, triglycerol, these are there. So I'll talk about all those enzymes that are there and in detail, and I do have a movie for you to look at it, so get like what's happening in terms. And as I just told you, when they come together, the gap is called synapse. It's called immunological synapse. And the whole response that sets in, we call immune response. Be more specific, it is called effector response. So that is what happens and set in. Now, before I go into detail, let me talk about some of the important intracellular events. It could be for both. It could be for CD4 T cell, A cell, B cell, because they're all lymphocytes. They all go through this process. They all go through this process. And sometime, depending upon what particular antigen or intracellular signaling we are talking about, it could occur within seconds, minutes or hours. It could be very fast. So by the end of the day, what happened is that these events basically want to take the signal from the cell membrane all the way to the nucleus, to the genes. And then genes will start expressing whatever is required. For example, you want cell to produce a particular cytokine, it has to come, message has to come to the headquarters, which is nucleus, and there has to be a gene expression. So we can have a protein synthesis. So all these intracellular events are important. And there are 3 lines of drugs which are in the market. Allergy drugs in the pipeline. Drugs for transplantation, drugs for cancer, drugs for autoimmunity. These are 4 or 5 very important things which are out there. And as I said earlier, that will be good background information for you to grasp this concept. Okay, now if you pay attention into the intracellular events in CD4 T cell activation, so as it say, for simplicity, it's not that simple as you see in this one. Okay, so we have taken many things out from here so that it's become easy for you. But just to give you an upshot, that and I usually say that the more intracellular pathways, the more enzymes, the more complication, the better is the prospect of pharmacist. So this will give you job guarantee. Because if had it been simple, we would have just need one pharmacist take care of that, dispense the drug. So it's good, it's a good job prospect for the pharmacist, that they keep on discovering new molecules, the new drugs are coming, so on and so forth. So that's what R&D development is there. And in R&D, it's not only pharmacists, physicians go, cell biology, molecular biology, they're all different fields that there. But you can see from here, some of things may look similar to you. So try to kind of, at least, even if I don't test you, even if I don't ask you, but out of curiosity, you would want to know how and where a particular drug is acting and what is going on in this particular immune response. Now, if you pay attention over here, this is something that may look familiar to you, right? In the red block over here, correct? Because what it shows is that there is MSC class 2 peptide, it talks to a TC4, there's a zeta chain. Remember when I talked about zeta chain, I said there are items expressed at the bottom in the intracytoplasmic tails of zeta chain. That's what you see over here. Now, when everything docks in, then all of a sudden, something happens that will send a current, that will send a message. So in this case, you can see from here, one of the things which is not mentioned on this one is basically, let me give you an overview. So there are enzymes in cytoplasm. Okay, one of the enzymes are kinases, right? And they are, as the name suggests, they are those enzymes, they will set the things in motion, like kinetics, move on. And some of them are phosphatases, and they have their very specific name. So one will put a phosphorus ligand, we call it phosphorylation of a particular enzyme. So this will set the activity. The other one will dephosphorylate. So if you pay attention over here, once the signal goes over here, and this is passed into the cytoplasm, then there are tyrosine kinases. We call it tyrosine kinases. So these tyrosine kinases, for example, in this case, you have FIN, these are the names of that, ZAP70. So these have to come in contact with each other, because remember that nothing will pass into the cytoplasm unless they come in contact with each other. So the very first signal will be phosphorylation of these moieties, items, and it will cause attachment of this FIN and ZAP and another molecule LIC, and this will then send a signal to some of the adaptor proteins which are just hung underneath the cell membrane. So these are very specific molecules. Okay? So you can see from here, these are adaptor proteins. And adapter proteins basically, I don't know whether you can read it from here because I cannot. From here. So you can see from here, these are basically enzymes which are there and they need to be activated. And then those of you who remember even production of some of the enzymes or some of the hormones, there is a sequence over here is pretty much a common sequence for many different molecules. For example, when I will discuss, I hope, asthma, and you will wonder why is it that the secretions become thick in patient of asthma, and how can we block these secretions? So these secretions are upregulated because activation of these molecules expressed over here. Okay, so PIP2 goes to PLC and then DAG. So this scheme over here, this one over here, apart from this immunological concept, this is something that is going to come over and over again, So I would advise you to look into that because it's just like your Krebs cycle or electron transmission chain, so on and so forth. Because many of the molecules, the drugs acting in cholera, for example, the drugs acting in asthma, they're all going to go and process into this sequence. So what you see over here is that there are tons of molecules, and they are RAS and RAC and IP3, and so again, you don't need to remember the name, but remember that they will lead to signals that are there to send a message to the cell as we call it a second signal, and you can see from here, one of them is calcineurin, activation of transcription factor. So the signal will lead on to activation of transcription factor. And transcription factors are such that unless and until they become activated, no gene expression will take place. Nothing will be synthesized in the nucleus because they have to make sure that these transcription factors enter into the nucleus of the cell. And there are hundreds of transcription factors, but I would at least want you to remember only 3 because we'll talk about that in detail. One is NF-kappa B, the other one is NF-AT, and then AP1. So these are 3 important transcription factors, and I'm pretty sure when you would have done pathophysiology, cancer, and why the cancer is caused, and so on and so forth, because you can see from here, there are 2 important drugs, as you can see, cyclosporin A and tacrolimus. So these are the 2 drugs available in the market, and we use them to prevent graft rejection. Because we give you a graft, you want this graft not to be rejected, so we give you 2. So these are anti-graft rejection drugs. So once we go into P2, P3, they will tell you cyclosporin A, tacrolimus, and they will want you to know that they basically act on calcineurin. So you should have a visual image, where is calcineurin and what is the function of calcineurin. Okay? So you can see from here that there is something over here that regulates. And we have hundreds of molecules, and the drugs in pipeline. I remember one of the students came to me about the project novel drugs. So these are the novel drugs. Penicillin is not a novel drug. Whatever drugs are given in your book, Murray, they are not novel drugs. The drugs related to intracellular pathways, immunology, they are the novel drugs because they are coming up. Okay? So this is what's happening, and most of the time, I would still encourage you, get used to these terms, adaptor protein, kinases, for example, MAP kinases, SARC kinases, phosphorylation, dephosphorylation, phosphatases, and transcription factors, because expectation is once we go into detail, you will pretty much have an idea as to what is going on. So you can see, we call it a cascade as well. So there's a cascade of initiation of events, okay? And then again, I just put over here some of them, but again, once we go in detail, I will talk about that in the next lecture. Let me show you a movie.